The moment a casket is sealed underground, a clock starts. A real, physical sequence of thermodynamic events that will, over the next century, disassemble everything that was once a person and redistribute it through the soil, the water table, the atmosphere, and eventually into other living things. Every stage follows the same law of physics, the second law of thermodynamics, running in the direction entropy always intended. The process begins from within. Your body, the one that carried groceries, that held someone's hand, that sat in a kitchen on a Tuesday morning drinking coffee, contains everything it needs for its own disassembly, and it starts using it within minutes. The iron in your blood traveled 13 billion years to reach your bone marrow. Where it goes next takes roughly a decade. That journey comes later. By the century mark, some of your atoms will be in the body of a child who has never heard your name. This is where you go. The first minutes, you might expect the disassembly to begin from outside bacteria, insects, the environment attacking an inert body. The physics runs the other direction. It starts from within, and it starts with electricity. Every cell in your body maintains a voltage across its membrane, roughly -70 millivolts on the inside relative to the outside. This membrane potential is maintained by sodium-potassium pumps protein machines embedded in the cell membrane that spend ATP, the cell's energy currency, to push 3 sodium ions out of the cell for every 2 potassium ions they pull in. The pump runs continuously. It runs against the concentration gradient, forcing ions in the direction they would not naturally flow, and this costs energy. Roughly 30% of the body's resting energy expenditure goes to maintaining these ion gradients. The voltage they create is what makes nerve conduction, muscle contraction, and cellular signaling possible. Without the pumps, the voltage collapses. Without the voltage, the cell cannot function. When oxygen delivery stops, when the heart ceases and blood flow ends, ATP generation halts within seconds. The mitochondria, which require oxygen to run the electron transport chain that generates ATP, go silent. Existing ATP reserves are consumed within minutes, and those reserves are all that stands between cellular order and cellular collapse, because every structure in every cell depends on continuous energy to maintain itself. Without ATP, the sodium-potassium pumps stop cycling. Sodium floods into the cells along its concentration gradient. Potassium leaks out. Calcium, normally maintained at vanishingly low concentrations inside the cell through energy-dependent pumping, enters uncontrolled through voltage-gated channels that can no longer be regulated. The membrane potential collapses from -70 millivolts to 0. The cell is electrically dead. Calcium influx triggers the next stage. Calcium, at high intracellular concentration, activates proteases, enzymes that cut proteins, including calpains, which begin severing the structural proteins that give the cell its shape. The cytoskeleton disintegrates. The cell's internal architecture, the scaffolding of actin filaments and microtubules that held everything in position, comes apart. And then the lysosomes rupture. Lysosomes are membrane-bound organelles that normally function as the cell's recycling centers, containing powerful digestive enzymes safely enclosed within their own membranes, breaking down waste material in controlled conditions. The lysosomal membrane, like every membrane in the cell, requires energy to maintain its integrity. Without ATP, the membrane destabilizes. The lysosomes burst, releasing their cargo, cathepsins, lipases, proteases, nucleases, directly into the cell interior. The enzymes that were contained for a lifetime are now loose inside the cell they were designed to maintain. The containment was the achievement. The release is what happens when the achievement can no longer be sustained. This is autolysis, self-digestion. The cell digests itself using its own machinery. No external agent is involved. Your own enzymes, freed from the containment that energy had maintained, begin dismantling the structures energy had built, the same structures that let you walk to the letterbox this morning, that let you feel the warmth of a cup in your hands, that let your eyes focus on a face you loved. The thermodynamics are precise. Every structure built during life required energy input to create and maintain. The proteins were folded against their thermodynamic preference, held in specific configurations by continuous energy expenditure. The lipid membranes were maintained against the entropy that would dissolve them. The ion gradients were pumped against diffusion. Remove the energy, and every one of these structures relaxes toward its thermodynamic ground state: unfolded proteins, disrupted membranes, equilibrated ion concentrations. Autolysis is a return to thermodynamic equilibrium. The second law was always pulling in this direction. Life was the temporary force holding it back. Everything that follows is the second law resuming what it was doing before you were born. Organs fail in order of their enzyme content. The pancreas goes first. An organ whose primary function is manufacturing digestive enzymes is, predictably, the first to be dismantled by digestive enzymes. The liver follows, rich in metabolic enzymes that now turn inward. The brain, with its high water content, roughly 78%, and its delicate lipid-rich membranes liquefies faster than muscle tissue, which has denser protein cross-linking that slows enzymatic access. Within 72 hours, the internal organs have lost structural integrity, taken apart from the inside, using only the tools carried in life. The first weeks. Now the bacteria arrive, but they were already there. Your body, in life, hosted roughly 38 trillion bacteria, a population approximately equal to the number of your own cells. The majority lived in your gut, specifically in the large intestine, in an anaerobic environment you maintained for them. Every meal you ate fed them too: the breakfast you had this morning, the dinner you cooked last night, the thousands of meals across decades that sustained both you and the ecosystem inside you. They digested components of your food you could not process yourself. They synthesized vitamins you needed. The relationship was mutualistic. The barrier that maintained it was the intestinal wall, a single layer of epithelial cells, held together by tight junctions, separating the bacterial ecosystem from the sterile tissue beyond. Autolysis dissolves the tight junctions. The intestinal wall loses integrity. The bacteria Clostridium perfringens, Bacteroides species, Bifidobacterium, encounter tissue that can no longer maintain barriers against them. The migration is outward, from the gut into the surrounding tissue, following the path that autolysis has opened. The bacteria are doing what they always did, metabolizing organic material using anaerobic fermentation pathways. The substrate has changed. The chemistry has not. Metabolic byproducts of this stage are specific and measurable. Bacterial proteases cleave proteins into amino acids, and the amino acids undergo decarboxylation, producing cadaverine from lysine and putrescine from ornithine. These diamines are the molecular signature of decomposition, generated by known enzymatic reactions from known substrates. Simultaneously, the bacteria generate gases, hydrogen, sulfide, methane, ammonia, that accumulate in the body cavities and inflate the tissue. The green discoloration that appears on the abdomen is hemoglobin being chemically altered. Hydrogen sulfide reacts with the iron in the heme group, forming sulfhemoglobin, a green pigment. Your blood's oxygen-carrying molecule is being converted by the waste gas of the bacteria you fed your entire life. Irony runs through this stage. The microbiome, the bacterial ecosystem you maintained, nourished, and depended on, is now recycling you, using the metabolic pathways it always used. The Clostridium species that fermented dietary fiber in your colon are now fermenting colonic tissue. The enzymatic machinery is identical. The substrate has shifted from food to host. The relationship was always chemical. The boundary that made it mutualistic was physical, a membrane. The membrane is gone. By the third month, the pace shifts: slower chemistry, larger scales, and in certain conditions, the body does something no one expects. It makes soap from its own fat. That chemistry comes shortly. Soft tissue progressively liquefies as bacterial enzymes and residual autolytic enzymes continue breaking proteins into amino acids and then into simpler nitrogen compounds, ammonia, ammonium, and eventually nitrate through soil bacterial processing. The rate at which this occurs is governed by chemical kinetics, specifically by the Arrhenius equation, which describes how reaction rates depend on temperature. For every 10 degrees Celsius increase in temperature, most biological reaction rates roughly double. A body buried in tropical soil at 30 degrees Celsius decomposes at approximately 4 times the rate of a body buried in temperate soil at 10 degrees. The physics sets the clock speed. The chemistry runs at whatever pace the temperature allows. What happens next depends on water. In wet, oxygen-poor environments, something specific happens to fat tissue. Triglycerides, the storage form of fat, consisting of glycerol bonded to 3 fatty acid chains, undergo hydrolysis. Water molecules break the ester bonds connecting the fatty acids to the glycerol backbone. The freed fatty acids, primarily palmitic acid and stearic acid, both long-chain saturated fats, react with calcium and magnesium ions from the surrounding tissue and soil, forming metallic soaps, calcium palmitate, magnesium stearate. The result is adiposarray, a waxy, grayish-white substance that is, in chemical terms, soap. The reaction is saponification, the same reaction that has been used to manufacture soap for thousands of years. Fat is being converted to soap using calcium from the body's own bones. Adiposity can preserve the external contour of a body for decades, sometimes centuries, because the metallic soaps are resistant to further bacterial degradation. The chemical transformation stabilizes what would otherwise continue dissolving. In dry environments, the physics runs differently. Evaporation removes water from the tissue faster than bacteria can consume it. Without water, the hydrolysis reactions that drive decomposition slow dramatically because water is a reactant, not merely a medium, in the enzymatic cleavage of peptide bonds and ester bonds. The tissue desiccates. Partial mummification results. The proteins denature and cross-link in the dehydrated state, forming a tough, leather-like material resistant to further enzymatic attack. Which path your body takes, liquefaction, saponification, or mummification, depends on the local physics, temperature setting, reaction rates, moisture-enabling hydrolysis, soil pH determining enzyme activity, and oxygen availability determining whether aerobic or anaerobic pathways dominate. The environment decides. The molecules obey. After the first year, a different material takes over the timeline. Once soft tissue is gone, consumed by bacteria, dissolved by enzymes, converted to adiposity, or desiccated beyond biological access, what remains is the skeleton. And the skeleton tells a different story, because it is made of different material operating on a different time scale. Bone is a composite material, roughly 70% crystalline mineral and 30% organic protein. The mineral component is hydroxyapatite, a calcium phosphate crystal, with the formula K10PO4 SEN OH2. The organic component is primarily type 1 collagen, a triple helix protein that provides bone with its flexibility and tensile strength, the way rebar provides reinforced concrete with resistance to bending. The mineral provides compressive strength. The combination of mineral rigidity and protein flexibility is what made your skeleton capable of bearing load while absorbing impact across a lifetime. Collagen degrades over years to decades, depending on soil conditions. Bacterial collagenases enzymes specifically evolved to cleave collagen's triple helix work slowly because collagen's tightly wound structure limits enzymatic access. Soil pH matters. Acidic soils accelerate collagen hydrolysis, neutral soils slow it. Temperature matters through the Arrhenius relationship. Warmer soils degrade collagen faster. Moisture matters because hydrolysis requires water. The collagen in bone, buried in cool, dry, neutral soil, can persist for centuries. In warm, wet, acidic soil, it may be gone within a decade. Mineral dissolution operates on a longer time scale, entirely. Hydroxyapatite dissolves through a process governed by its solubility product, the equilibrium constant that describes how readily the crystal dissociates into its component ions in solution. In acidic conditions, soil pH below 6, the hydroxyl groups in the crystal lattice are protonated, destabilizing the structure and accelerating dissolution. In neutral or alkaline conditions, pH 7 and above, the crystal is thermodynamically stable and dissolution slows to geological timescales. Teeth 96% mineral, encased in enamel, that is the hardest biological substance your body ever manufactured, persist the longest. Tooth enamel is almost pure hydroxyapatite, with larger, more perfect crystals and fewer defects than bone mineral, giving it lower solubility and greater resistance to dissolution. Teeth can survive thousands of years. They are the last recognizable remnant of the physical structure that was you. Teeth that bit into an apple. Teeth that smiled in a photograph. Lasting longer than anything else the body built because the crystal was more perfect, the bonds were stronger, and the physics of dissolution runs slower on a denser lattice. If someone visits your grave 10 years after burial, the skeleton is likely still present, surrounded by the chemical remnants of everything else. The soft tissue has been converted to soil compounds. The bacteria have consumed what they could access. The skeleton remains because mineral dissolution is orders of magnitude slower than protein degradation, because the activation energy required to break ionic bonds in a crystal lattice is far higher than the activation energy required to hydrolyze a peptide bond. The physics of bond strength determines the order of disappearance. Weakest bonds first, strongest bonds last. Above the grave, the soil chemistry has changed. The ground directly above a burial site is measurably enriched in nitrogen, phosphorus, and potassium compared to surrounding soil, a nutrient signature detectable by soil analysis for decades after burial. The grass above a grave often grows differently, greener, denser, taller, because the nutrients released by decomposition have entered the root zone. Anyone who has walked through an old cemetery and noticed that the ground above the graves seems more alive than the ground between them was observing nutrient cycling in real time. The dead are feeding the living. Thermodynamics guarantees it. From the 10th year onward, the story changes direction from dissolution to incorporation. This is the stage I think about most because the physics stops feeling like an ending and starts feeling like a transfer. Nitrogen first. Your muscles contained roughly 1.5 kilograms of nitrogen, bound in protein in the amino acids that form the actin and myosin filaments that powered every movement you made across a lifetime. Every step you took, every hand you raised, every breath that expanded your ribs against gravity, the nitrogen was there for all of it, locked into the protein architecture that made movement possible. As bacterial decomposition breaks those proteins into amino acids and then into ammonia and ammonium, the nitrogen enters the soil solution. Soil nitrifying bacteria, Nitrosomonas species, oxidize ammonium to nitrite in an energy-yielding reaction that sustains the bacteria's own metabolism. Nitrobacter species then oxidize nitrite to nitrate, again energy-yielding. The bacteria are running these transformations because the oxidation of reduced nitrogen compounds releases free energy they can capture through chemolithotrophy. The nitrogen cycle runs on thermodynamics, each step releasing energy, each organism capturing enough to sustain itself while passing the nitrogen forward in a more oxidized form. Nitrate, the final bacterial product, is the form of nitrogen most readily absorbed by plant roots. Your muscle nitrogen, converted through 2 bacterial oxidation steps, is taken up by roots through specific nitrate transporter proteins and incorporated into new amino acids, new proteins, new plant tissue. The nitrogen that powered your arms is now powering the growth of a tree. The nitrogen does not carry a memory. It carries a charge. It is the same element in a different oxidation state, in a different organism, performing an analogous function, building structure, enabling growth, doing what nitrogen does. Phosphorus and calcium, both locked in the hydroxyapatite crystal lattice of the skeleton, follow slower paths into the same destination. As bone mineral dissolves over decades, phosphate and calcium ions enter the soil solution and are taken up by plant roots. Phosphorus is often the limiting nutrient in terrestrial ecosystems, the element in shortest supply relative to biological demand, and your body's phosphorus, released from a kilogram of bone, represents enough local nutrient input to sustain a medium-sized tree for a decade. The phosphate enters new ATP molecules, new DNA strands, new phospholipid membranes. The calcium enters new cell walls, new signaling pathways. The minerals that gave your femur its compressive strength are giving rigidity and structure to the living tissue growing above your grave. Carbon follows the atmosphere. Bacterial metabolism of your tissue converts organic carbon to carbon dioxide through cellular respiration, the reverse of the photosynthesis that originally captured it. Every carbon atom in your body was once a CO2 molecule in the atmosphere, captured by a plant, fixed into glucose using the energy of sunlight, and passed through a food chain until it reached you. Decomposition reverses the process. The CO2 enters the soil atmosphere and eventually the air above. Atmospheric mixing distributes CO2 globally within roughly a year. The carbon that was part of your liver could be inhaled by a plant on another continent within 12 months of its release. A plant somewhere, a tree in a forest you never visited, a crop in a field you never saw, captures that carbon dioxide molecule through the Calvin cycle, reduces it using electrons stripped from water, and builds it into a new sugar molecule. That sugar becomes cellulose in a cell wall, starch in a root, lignin in bark. Some of it is eaten. Some of those carbon atoms, atoms that were part of the protein in your heart muscle, or the lipid in your brain membranes, or the collagen in your skin, are now part of another organism. The redistribution is governed by atmospheric mixing, by photosynthetic rates, by food web dynamics. The carbon does not know where it has been. It follows the thermodynamics, cycling between the atmosphere and the biosphere with a turnover time measured in years through a cycle that is run unbroken for 3.5 billion years. By the century mark, the redistribution is extensive. Your atoms are dispersed in the soil, in groundwater, in the atmosphere, in the tissue of plants that grew in the soil above you, in the bodies of organisms that consumed those plants. The distribution cannot be mapped because it is governed by diffusion, by water transport, by atmospheric circulation, by food web dynamic systems that distribute material across scales too large and too complex for any tracking. But the distribution is thermodynamically inevitable. Entropy, the tendency of concentrated systems to disperse, of ordered arrangements to spread into less ordered ones, guarantees this outcome. The second law does not erase you, it distributes you. The matter that was concentrated in one place, in one form, for one lifetime, returns to the dispersed state from which it was originally assembled, because that is the reframe this entire sequence has been building toward. The process described above, autolysis, bacterial decomposition, saponification, mineral dissolution, nutrient cycling, runs in one direction, from concentrated and ordered to dispersed and distributed. Life ran it in the other direction. Life took dispersed atoms, carbon from atmospheric CO2, nitrogen from soil nitrate, calcium from dissolved minerals in water, phosphorus from soil solution, iron from geological weathering, and concentrated them into one specific, improbable, highly ordered arrangement, you. The energy to do this came from the sun, captured by photosynthesis, passed through food chains, used by your metabolism to build and maintain the structures that made you a person, proteins folded into specific configurations, membranes assembled against entropy, ion gradients pumped against diffusion, bone mineralized into crystal lattice. All of it was entropy held at bay by continuous energy input. Every atom that was concentrated into you was previously part of something else. The calcium in your bones right now was in the ocean, once dissolved as calcium ions, precipitated into the shells of marine organisms millions of years ago, deposited as limestone on the seabed, uplifted into mountains by tectonic forces, weathered back into solution by rain over millennia, taken up by the plants and animals that formed the food chain you ate from. That calcium has been through hundreds of organisms before you. You are one stop on a journey that began when the element was first forged in a dying star and will continue long after your skeleton has dissolved. Nitrogen follows a similar path backward. The nitrogen in your muscles was atmospheric N2, the inert gas that makes up 78% of the air before soil bacteria broke the triple bond between its two atoms and fixed it into ammonium, a form plants could absorb and you could eat. Breaking that triple bond requires enormous energy. It is one of the strongest bonds in chemistry, requiring 945 kilojoules per mole to cleave. The bacteria that fix atmospheric nitrogen use an enzyme called nitrogenase that consumes 16 ATP molecules per nitrogen molecule. Fixed a massive energetic investment that the bacteria make because reduced nitrogen is the currency of biological growth. The nitrogen in your biceps was fixed from the atmosphere by a bacterium in soil, absorbed by a plant, eaten by you, built into muscle protein, and will be returned to the soil by the bacteria that decompose you, where it will be fixed, absorbed, and built again. The cycle has no beginning and no end. It has iterations. You were one. Burial returns the atoms to the pool they came from. The iron in your blood, every atom of it was forged inside a star that exhausted its hydrogen fuel and fused progressively heavier elements in its core. Hydrogen fused to helium, helium fused to carbon, carbon to neon, neon to oxygen, oxygen to silicon, silicon to iron. Iron is the endpoint, the most stable nucleus, the element with the highest binding energy per nucleon, the point beyond which fusion no longer releases energy but consumes it. The star reached iron and could fuse no further. It collapsed under its own gravity and exploded a supernova, scattering those iron atoms across light years of space. The atoms drifted through interstellar medium for millions of years, were swept into the gas cloud that condensed into our solar system 4.5 billion years ago, accreted into the planet, were weathered out of rock by water over geological time, entered the soil solution, were taken up by plants, eaten by animals, passed through food chains, and arrived in your bone marrow, where they were incorporated into hemoglobin molecules that carried oxygen through your arteries for every second of your life. That iron, forged in a star, scattered by an explosion, drifting through space, compressed into a planet, dissolved by rain, absorbed by roots, eaten, digested, and built into the molecule that kept you alive, is now returning to the soil. It will be taken up by the next organism that needs it. The journey from star to you took roughly 13 billion years. The journey from you to the next organism will take roughly a decade. The iron does not distinguish between hosts. It follows the chemistry. That disproportion stays with me 13 billion years to arrive, a decade to move on. The universe spent most of its history delivering those atoms to you. Your contribution to the journey is a single, brief stop. You are one iteration of a cycle that has run for 4 billion years on this planet, and for the elements forged in stars, for roughly 13 billion years. The cycle does not stop when the iteration ends. The matter goes on, into the next root, the next leaf, the next organism, the next body, indifferent to names, indifferent to identity, subject only to the physics that has always governed everything. Consider what this means about the body you are occupying right now. The atoms you are made of were part of other organisms before you. The calcium in your femur was in the bones of another animal, perhaps many other animals before geological and biological cycling delivered it to the food chain that fed you. The nitrogen in your muscles was in the muscles of other organisms, passed through soil, through bacteria, through plants, through the food on your plate. The carbon in your brain was atmospheric CO2 that a plant captured, that an animal ate, that you ate, that your metabolism built into the lipid membranes of your neurons. Every atom in your body is borrowed. Every atom was somewhere else before it was you, doing something else, in something else, and will be somewhere else after. What you think of as your body is a temporary arrangement, a specific configuration of atoms held in place by continuous energy input, persisting for a few decades, and then returning to the pool when the energy stops. The configuration is unique. The atoms are not. They have been through this before, and they will go through it again. The only thing that changes between iterations is the arrangement, the specific, improbable, unrepeatable pattern that was you. The pattern ends. The matter continues. And the next pattern, built from the same atoms, will be equally specific, equally improbable, and equally temporary. A casket sealed, a clock started. From the first enzyme released in minutes to the last mineral dissolved across decades, each stage governed by specific chemistry, each rate set by specific physics, each product entering a cycle that was running before you were born and will continue after every trace of your structure has been redistributed into the living systems that surround the place where you were laid down. The second law brought you here. Every atom in your body was assembled from dispersed material into an ordered structure by the continuous input of energy captured from the sun. The second law takes you back. Every atom returns to the dispersed state from which it was gathered into soil, into water, into atmosphere, into the tissue of organisms that will never know where their nitrogen came from or whose bones supplied their calcium. And the atoms that will carry someone else's oxygen, build someone else's bones, power someone else's muscles, those atoms are yours, for now, on loan from a universe that has always recycled everything it makes.